Hi, folks, and welcome to another edition of Zog Science. Today, we're going to be talking about reproduction. So, just a little general review of some of the things we've talked about before. Where there are two modes of reproduction. We've got asexual and sexual reproduction. Recall that asexual reproduction generally involves mitosis, binary fission, something like that, where you have one parent that is turning into two daughters. And then you have sexual reproduction, which is where you're going to have two parents combining together to form well, not combining together, but going to be leading two offspring and is going to generally involve meiosis somewhere along the way because we need to produce haploid cells that can then combine together to become diploid. Um, got a couple diagrams down here just as a reminder. Also remember, we've got some different, kind of general modes for animals, plants, and fungi, and those are the three groups that we're going to be talking about today. So let's start off with fungi. Fungi are generally haploid, however, they do become diploid in order to reproduce. The reproductive structure is what we think of when we see mushrooms. That is actually just the reproductive structure. The rest of the fungi is generally hidden from view in whatever substrate it is breaking down. The general fungal life cycle, you can see that here. In general, you spend most of the time as a haploid organism. It can become heterokaryotic, and what that means is that it is both haploid and diploid. Different stages of it are, and so then you go in and you go through this process of having this heterokaryotic stage that then can then produce a zygote, which will go through meiosis that will eventually produce spores that will kind of go back into the normal rest of the life cycle. So let's take a look at a couple of examples. This is a Ascus fungi, and so what it does is it produces these spores. These spores grow into the mycelia, which are kind of like the hyphae. It's just a general plant fungal body, not plant body, fungal body. And what happens is that that fungal body will produce two different mating types, the plus and the minus. And if the plus and the minus ever come in contact with one another, then it will produce the ascus, which is where the zygote will actually be produced. That zygote will then eventually go through meiosis, and then the meiosis then turns into these spores. The spores are then dispersed, and it turns back into the mycelia, which are kind of doing the normal fungal activities. The other type you can have is what is known as a basidiocarp. I'm sorry, a basidius, which in this case it has a basidiocarp. Anyways, they have little gills, and inside those gills, that is where the zygote is being produced. The zygote again is then released, is going through the process of meiosis, and then the spores are being released. They're forming the mycelia. Mycelia again produce the plus and minus. If they come into contact with one another, they combine, and so now you get the heterokaryotic state where you've got some of it that's haploid, some of it that's diploid. The diploid section again goes through and produces the diploid zygote, which then continues the process forward. So fungi have kind of a weird life cycle in terms of what we tend to think of as being normal. All right, so let's talk about animals now. While fungi are mostly haploid, animals are mostly diploid and only sometimes are haploid. The animals usually are going to produce via sexual reproduction. However, some animals can do asexual reproduction. These are earthworms that are reproducing sexually. Earthworms are hermaphrodites, which is why they're both going to be passing sperm to the other. These right here are sea anemones. Sea anemones are animals, and they can reproduce asexually if needed. A little kind of weird thing is parthenogenesis. Parthenogenesis is the development of unfertilized eggs. This is where mating pairs swap playing the role of males and females. So these are technically both females. However, one of them is going to be male-like. It's going to exhibit male type behaviors, and that sexual behavior that the male is exhibiting is going to be required in order to stimulate ovulation. So you need something to act male-like in order to happen, but you don't actually have to have swapping of sperm occur. Now, obviously, this is going to result in not quite as much sexual diversity. However, it also, is a little bit easier and a little less energy intensive, which can be an advantage as well, depending on what type of environment you are in. Some other adaptations, you can either have external or internal fertilization. Obviously, external fertilization is going to put your babies at a lot more risk, as opposed to internal fertilization. External fertilizing organisms tend to release a lot more eggs, and tend to have a lot more children because more of them are going to be eaten. Other things are the amount of parental care. So, whether or not you take care of your babies or you just kind of leave them behind. Obviously, taking care of them takes a lot more energy, but however, it also is going to increase the likelihood that they are going to be able to survive. So let's talk a little bit about human reproduction. In human reproduction, the sperm is going to fertilize the egg. Both of those are haploid. They're produced by meiosis, and they're going to produce the zygote, which is diploid, and that is going to occur inside the oviduct. So the two of them are going to come together, and then the blastocyst is going to implant in the uterine lining. The blastocyst is what forms after a few cell divisions are going to occur. So it's not just a couple of cells in there. It's probably hundreds of cells at this point when it's actually going to be implanting. It probably takes around 24 to 72 hours for the zygote to move down the rest of the oviduct. And we'll talk a little bit more about the whole development side of this in our next topic, which is development. And then once it implants in the uterus, the baby is going to develop and grow. The reproductive systems for males and females are both similar and different at the same time. They both have a place where that is going to be producing the actual sex cells. So that is in the males, that is the testes, where the sperm are being produced, and in the females, it is the ovaries. And then there is, kind of a duct. For the males, that is the vas deferens. For the females, that is the oviduct that is going to be transporting the eggs down into the uterus. And then both of them, have a way to expel those sex cells. Eventually, obviously, males, it's different because the goal of the male reproductive system is to put sperm inside the female reproductive system. And that's kind of the main difference between males and females, especially in terms of reproduction. When you look across species, is that males are the ones that or the male sex cells are the cells that are going to be mobile. They tend to move around, they're going somewhere, whereas the female sex cells, they are not necessarily going anywhere. They are staying put, and they are being met by the sperm cells. If we take a look at the female reproductive system, we've talked about the endometrium before in terms of the menstrual cycle, and this is just to kind of give you an idea of where that actually is. The endometrium is inside the uterus. It's that inner lining of the actual uterus. It's not the uterine wall, and it is going to, it's gonna be there, but it builds up and decreases based on whatever the menstrual cycle is. And we'll get into that a little bit more again once we get into development. We'll talk a little bit about what happens when implantation actually occurs. But let's talk a little bit about how the sperm and eggs are actually produced. So what happens is that inside the testes, there are these cells that are called spermatogonium, and the spermatogonium are what are the precursors to actual sperm. So they go through mitotic division, a couple of mitotic division, where they turn into a spermatocyte, and then after a spermatocyte, they go through meiosis, and they produce the secondary spermatocyte, and then they go through mitosis again. I'm sorry, meiosis 2, and that's when they actually become the early spermatids. And then there's some differentiation that occurs. There are cells that provide them with specific nutrients, and they actually are then going to turn into the sperm cells. They're going to express certain genes that are going to allow them to turn into those sperm cells. And from there, they will then go into the vas deferens and eventually to be released through the urethra. Now, males are going to be producing hundreds of millions of sperm each day, and the ones that are not released are either absorbed or they are gotten rid of through something like a wet dream, things like that. is going to begin in puberty and it ends at death. So males can be have vitality throughout the entirety of their, I'm sorry, not vitality, fertility throughout the entirety of their lives. Although the sperm tend to not be quite as capable towards the end of their life, although that is to some extent a genetic trait. And you get 4 sperm per every 1 spermatogonia that you start off with. Now, contrast that, we'll talk about oogenesis. Instead of it being hundreds of millions a day, it's only 1 a month. And it does as well begin at puberty. There is some question as to whether or not a female is born with all of her eggs when she is first birthed. To the best of our knowledge, that's what we believe. However, we have found in some animals that follicles are being produced and are dividing as the animal is maturing and growing. So it's not 100% sure, but at this point, we believe that a female is born with all of the follicles that she is going to have. And so what happens is that the follicle will, arrest development before the end of meiosis one. I'm sorry, before meiosis one begins. So the development is arrested in prophase of meiosis one. Then once that follicle is actually goes through the process of ovulation and it releases the oocyte, that is when the rest of meiosis one occurs and leads to the onset of meiosis two. Now, when that happens, the main difference between sperm and egg is that the egg is much larger than the sperm, and the egg is what houses all of the cellular machinery that is going to be used for the new zygote. So as the egg is going through its development, it's not evenly splitting. What it's doing is it's just getting rid of half of the genetic material and maintaining and keeping on to all of the other of the cellular machinery. That other half of the material is what is known as a polar body. And so as this process occurs, you get the formation of 2 polar bodies and the formation of your single mature ovum. That mature ovum doesn't go through the process of meiosis 2 until the sperm actually fuses with the egg, which is kind of an interesting thought. But when that sperm fuses with the egg, that's when it actually goes through the process of meiosis 2 and creates that fully haploid cell that will then be fertilized, and the 2 pieces of genetic information will come together. So I think that's a pretty interesting thing. The process of oogenesis will end at menopause. Females do not continue producing follicles for the and releasing oocytes and going through ovulation for the entirety of their lives. Menopause generally occurs somewhere between ages 40 and 50. No one's exactly sure why this happens, what the evolutionary benefits are. It could be that it encourages females to take care of the children that they already have, as opposed to continuously having children, which allows them to, provide more for the children that they already have. But again, this is all speculation. No one knows exactly why menopause occurs. There are a lot of different types of contraception that you can use that can help prevent the fertilization from taking place. Obviously, there's abstinence that is 100% effective. Some other ones that we haven't really talked about before in this class, at least, you can have vasectomy, which is where you cut the vas deferens, which means that the sperm are produced, but they never actually are able to leave the body. We've talked about birth control pills. They're hormonal controls of the female cycle. You can use a condom, which will obviously prevent the sperm from ever getting to the egg. We can have tubal ligation, which is where the oviducts are blocked off, usually by some sort of tying or cutting. You can use spermicides or some sort of diaphragm cervical cap that keeps the sperm inside the vagina but does not allow them to move forward. Or you can use a morning after pill or an IUD. An IUD is kind of interesting. It's a device that is inserted into the uterus that prevents the egg cell from implanting, and so therefore, if it's fertilized, it's going to have a much harder time implanting. So, anyways, I think that's it for animal reproduction. Let's talk about plant reproduction. Plants have this interesting alternation of generations where they can have both the diploid and the haploid organisms, similar to fungi. Plant reproduction can either be asexual or sexual. These are a grove of aspen trees, and these are all linked to one another. They are all actually the same organism. They're exactly genetically identical to one another. They're just growing next to one another, which is pretty interesting. However, most plants do sexual reproduction using some sort of pollinator, and we'll obviously talk a lot about that. Evolutionary trends in plant reproduction. We start off with mosses and other non-vascular plants having a dominant gametophyte, which is the haploid stage, and a reduced sporophyte that is dependent upon the gametophyte for nutrition. As we move forward, the sporophyte becomes dominant in vascular plants, while the gametophyte is gradually becoming reduced. Although for the seedless plants, they do have a free living gametophyte that can survive on its own. However, by the time you get up to your gymnosperms and your angiosperms, the gametophyte is very much reduced. Usually it's microscopic. You can't really see it very much. And the sporophyte is really dominant over it because the gametophyte is completely dependent upon it. So that's just kind of the overall trend, is that we start off with a haploid dominant and we end up with the diploid being dominant. So for gymnosperms, don't really know why that bullet point is there, but it's not that important. Gymnosperms, remember, those are your cones. There are male and female cones, both located on the same trees. They can have self-fertilization that can occur. And what happens is that the pollen grains, they land on the female cone, and the pollen will be able to fuse with it and create a diploid seed, which will then develop and is released, which can then grow into seeding, and so on and so forth. Angiosperms do things a little bit differently because they have flowers. The flowers have both male and female parts. They do their best to try to reduce self-fertilization. We'll talk about that in a little bit. But basically, you've got the male parts, which are the anther, and the female parts, which are the carpel. And the anther is going to be producing your sperm. And it actually takes 2 sperm in order to fertilize the egg. And again, we'll talk a little bit about that in just a moment. But the sperm are contained within the each pollen grain. So it's not like it takes 2 pollen grains. It only takes 1 pollen grain. But it takes 2 sperm, which is a little different from animals. And just like with animals, the sperm fertilizes the egg, becomes the zygote, and then it grows inside a seed. The seed is then released and is able to grow if it's in the right conditions. So let's talk a little bit about flower anatomy. Sorry, I may have misspoken. The anther is the pollen-producing part of the male, while the filament is kind of it's like a little mini stem, and together they are known as the stamen. The carpel is the totality of the female part. The stigma is at the top, which is what receives the pollen grain. The style is the tube that leads down to the ovary, where the actual eggs, or what they're called ovules in this case, are located. You've got petals, which are the colorful flowers that help to attract pollinators. The sepals are the green leaves. Now, there are 2 different types of angiosperms. You've got monocots and dicots, or monoecious and dioecious, and we'll talk about some of those differences a little later on. Gamete production, it's not super important that you understand exactly how these are produced. We talked about this a little bit before. Sperm leave the flowers, whereas the eggs do not. The eggs stay inside. It's the same thing we talked about with animals. Sperm are smaller than eggs, which is the same thing for animals. The polar nuclei, which are found inside the ovules, this is kind of interesting. They're going to fuse with the first pollen sperm that is able to come down. This is that double fertilization idea. And they're going to produce the endosperm, which is then going to, inside the seed, be the food for the egg cell. So when you're eating something like a peanut, you're eating the endosperm, which was produced from a peanut sperm cell that fused with the polar nuclei. And then the egg is going to fuse with the second sperm, and that's what actually produces the zygote that is going to be growing into the new plant. So this is that idea of that double fertilization. You've got the pollen grain, which creates a pollen tube that goes down the style that contains two sperm. The two sperm are then going to fertilize the zygote and fertilize the endosperm so that you get both the production of the new plant as well as the production of enough food for that plant to be able to grow once the seed finds appropriate territory. Now, attracting pollinators is a huge deal for plants because they need to transport their pollen around. We can either have pollination by wind, which is an abiotic factor. A little bit of ecology, remember from review from freshman bio. You can have either pollination by bees. Notice that under UV light, there's kind of a circle area around um, where all of the anthers are, which is good because that's going to help draw in the bees. Bees are able to detect ultraviolet light, which we are not. So you draw them in, and they are able to help. They're able to pick up a lot of pollen and then transfer it to other flowers, and hopefully they're coming into contact with the carpel and helping to fertilize. There's other adaptations. So you've got, either pollination by moths and butterflies. You can have pollination by flies. You can have pollination by birds and by bats, right? It's all about, being co-evolved. Remember we talked about this before, right? This is co-evolution. They've evolved together. They have specific traits that they are attracted to. They don't attract other insects. They don't attract other animals. They only attract the ones that they have evolved with. And by they, I mean now, ways to prevent self-pollination. Dioecious plants, those dicots have male and female flowers, so they're never together, which means that it's much less likely for them to actually fertilize each other. Also, the arrangement of the styles and the stamens, right? So if the stamens are below the style, that helps to reduce the opportunity for the pollen to actually fall down and self-fertilize on the style. So a couple different ways to be able to take care of that. So seeds. Seeds are good because they help with dispersal. They resist desiccation, which means that they don't dry out very easily. They can also be dormant for a long period of time until they actually, get to the correct conditions, right? So you need enough water, good soil, things like that. You can also have fruit being formed on your angiosperms. Those fruits are good for dispersal, so they can attract animals to eat the fruits, which will then they'll then carry those with them, and then they will excrete them in other locations. They also provide protection, so you can have the fruit taste bad early in development while the seeds are still developing, and then once the seeds have developed properly, now the fruit releases aromas and things like that that attract animals or insects to come eat them. So different ways of dispersing seeds. You can either have winged seeds, the winged fruits of the maple. You can have kind of the dandelion parachutes. Tumbleweeds are there for dispersing seeds. Water, coconuts. That's the whole idea of a coconut is to be able to float in the water. You can have dispersed by animals. We talked about this, right? Seeds can be carried around by insects. You can have those kind of stickers that stick to fur that you can then carry around and eventually maybe knock off. Dispersal in the feces, and also, you can have things like squirrels taking seeds or fruits and burying them for later on. They probably don't remember all of them, and so they are able to eventually grow up. Germination. Germination takes place where the seed is buried underwater in the right conditions, usually with enough water. It's going to begin to germinate. It's going to send out some roots. Those roots are going to go down into the ground. And if it's a dicot, that is then going to pull the cotyledon, which is what sets apart a monocot from a dicot. Dicots have cotyledons, two cotyledons, which are the leaves kind of that form on the inside. You can see them right here. These are the either side. So when you're eating a peanut and you've got the kind of the two halves, those are your dicotyledons, the two cotyledons. And those grow into kind of the first leaves, and then you get leaves that grow on top of that. If it's a monocot, the monocot sends roots down, and it also sends the first leaf up, and that is the monocot. It's single leaf. So kind of interesting the differences between the two. You do need to know, I would say you need to know the major differences between the monocots and the dicots. Another word for dicot is eudicot, but dicot's in the word, so it's okay. Monocots have the one cotyledon, whereas the dicots have two. Monocots usually have parallel veins, whereas the veins of a eudicot are usually net-like. I think that those two and the flowers. So monocots tend to have floral organs usually in multiples of three, so either 3, 6, 9, something like that. Whereas for a eudicot, the floral organs, so the flowers usually have multiples of 4 or 5. And I would say that those are probably the three characteristics that you need to know the most. The taproot system is kind of important, but not really so much. We've talked about vascular tissue, but I'm not going to expect you to be able to know all that stuff. So let's just say that we know the embryos, the leaf venation, and the flowers, and we'll call it good. Is that good? All right. Well, thanks for watching this time. Next time, we've talked about reproduction now. Next time, we'll talk about development. So thanks for watching, and hopefully I'll see you next time.